Hello friends, today we are going to discuss about semi-conservative nature of DNA replication. We know that DNA is the genetic material of most of the currently known living organisms. Some exceptions are few viruses which have RNA as the genetic material. We have already discussed experiments performed by Frederick Griffith, Avery MacLeod and McCarthy and Hershey and Chase, which led to the conclusion that DNA is the genetic material. You can refer my video, the link is given in the description box. So now a genetic material should have following characteristics or some properties. First, it should be able to replicate, that is make exact copy of itself. It should be stable, structurally as well as chemically, and should be able to mutate or undergo changes in order to evolve slowly to survive in the changing environment. Francis Crick explains central dogma. DNA makes its copy with the help of replication. Also, DNA undergoes transcription to form messenger RNA. Then messenger RNA through translation forms protein. So, DNA sequence codes for messenger RNA and messenger RNA codes for protein sequence. This is known as central dogma. We will discuss replication, transcription and translation in detail in separate videos. Today, we will focus on semi-conservative nature of DNA replication. So now what is DNA replication? Replication is a process by which cell copies its DNA or produce two identical copies of DNA prior to cell division. Hence, each new cell receives a copy of DNA. This is important for inheritance. Earlier, three hypotheses were proposed for DNA replication. First is conservative mode. An entirely new DNA is synthesized from the original DNA, which acts as a template. So, one will be original parental DNA and other totally new DNA. Second is semi-conservative mode. Each new DNA molecule consists of newly synthesized strand and one template strand, that is original strand. Semi-conservative literal meaning is half conserved. So here one of the template is conserved, that is it is old one, and the second is newly synthesized strand. Then the dispersive mode. Some parts of original double helix are conserved and some are newly synthesized. Hence, daughter DNA molecules consist of part of template and partly newly synthesized DNA. Let us understand this in detail with the help of figure. This is the original DNA. If the mode of replication is conservative, then the entire old DNA or the original DNA serves as the template and the new DNA molecule is synthesized. And density gradient centrifugation will give two bands. This is of the template DNA and this is the newly synthesized DNA. In the semi-conservative mode of replication, this is the original DNA, the two strands separates. Now each strand serves as the template and the new strand is synthesized on the basis of the complementary base pairing, and density gradient centrifugation will give only one band. Now in the dispersive mode of replication, some parts of the DNA are the original double helix and some are newly synthesized. So some parts are conserved or the older ones and some are newly synthesized. And the density gradient centrifugation will give this type of the band. Later, semi-conservative mode of replication was confirmed experimentally. This is possible due to the complementary nature of two strands of DNA as proposed by Watson and Crick. They proposed double helical model where adenine always pairs with thymine by double hydrogen bond and guanine always pairs with cytosine by triple hydrogen bond. We have already discussed structure of double-stranded DNA. You can refer my video. The link is given in the description box. Two strands are antiparallel and hydrogen bond is present between the complementary base pairs. Hence can be separated easily because it is easy to break hydrogen bond. Each old strand of the double helix serves as the template for the synthesis of the new strand. Hence, two copies of the DNA molecules are produced. Each has one original strand and one new strand. And both the copies of DNA are identical. So, among two strands of DNA, one conserved and the other is synthesized according to the complementary base pairing. Hence, it is called as semi-conservative method or literally half conserved, because one of the strand, which is the template strand, is the old strand and it is conserved, and the other strand is newly synthesized. This mode of replication was experimentally proved by Messelson and Stahl in 1958. Now let us understand this with the help of this diagram. So this is the original DNA molecule. There are two strands. This is the original template. During the replication of the DNA, the two strands separate due to the breaking of this hydrogen bond between the complementary base pairs. Now, each of the older strands of the DNA serves as the template and the new strands are synthesized on the basis of the complementary base pairing. This is the one copy of the DNA and this is the second copy of the DNA. And in each copies, one strand is the template strand or the older one and the other is the new strand. So, half of the strand is conserved. Hence, it is known as semi-conservative method of replication. So, if we see here, this is the old strand and this is the new strand that is synthesized according to the complementary base pairing. Adenine always pairs with thymine and guanine pairs with cytosine. Similarly, this is the old strand and this is the new strand. Here also it is synthesized on the basis of the complementary base pairing. So there are two copies of the DNA. This is the one copy, this is the second copy. In the first copy, the old strand, it is identical to the new strand of the second copy. Similarly, the new strand of the first copy is identical to the old strand of the second copy. So, these two copies of the DNA, these are identical because of the complementary base pairing. Now, let us see Messelson and Stahl's experiment that was done in 1958, which proved semi-conservative mode of replication. E. coli bacterial cells were taken and they were grown in a medium containing 15N isotope. Till all the DNA was labeled with this 15N because DNA contains nitrogenous base. Then these cells were transferred to medium containing 14N and it was grown for few generations. And then it was analyzed by density gradient centrifugation. So what it was observed that after one generation, when it was grown in the 14N medium, all DNA had density of 15N and 14N. And after the two generations, it was observed that half of the DNA has the density of 15N and 14N and other half of the DNA has the density of 14N and 14N. Also, it was observed that fraction of 14 and 14N DNA or the frequency or the percentage of 14 and 14N DNA increases with every further generation compared to the DNA containing 15 and 14N. Now, let us understand this in detail with the help of this figure. This is the DNA of the E. coli that was grown in medium containing 15N nitrogen. Hence, both the strands are 15 and 15N. It was found out by the density gradient centrifugation, which gave the heavy band 15 and 15N. Then bacteria was grown in medium containing 14N. So the two DNA molecules that were synthesized, one strand was 15N and one was 14N in both the DNA molecules. And by the centrifugation, hybrid band containing 15N and 14N both was obtained. This is possible only if two strands of the original DNA containing the 15N separates, and then using this older strand as the template, new strand is synthesized by the complementary base pairing, and because it is grown in 14N medium, so this new strand is labeled by 14N, and hence the two strands of the DNA are 15N and 14N. Then in the next generation, if it is continued to grow in 14N, then half of the DNA was hybrid containing one 15N strand and one 14N strand. And half of the DNA had two strands labeled with 14 and 14N. And the density gradient centrifugation gave one hybrid band and one light band. This is also possible if the two strands of this generation separates. Then one has the 15N labeled strand. This serves as the template, new strand is synthesized, which is 14N. The other strand is 14N, which serves as the template, and then new strand is synthesized using the complementary base pairing, and it is 14N. Hence, half of the DNA is hybrid, 15 and 14, and half has the two strands 14 and 14N. If it is grown for further generation in the same 14N medium, then the fraction of the DNA containing 14 and 14N in both the strands will gradually increase, or the percentage of the DNA having the 14 and 14N strand will increase. And this proves the semi-conservative mode of DNA. That is, one of the strand is conserved and the other is newly synthesized strand. So there are certain applications of semi-conservative replication. Let us discuss them one by one. It is fast and accurate method of DNA replication. It allows easy repair of DNA strands because old and new strands are methylated at different times as they are synthesized at different times. Hence, before methylation, new strand can be corrected for any error as proofreading is possible. Also, certain areas of newly synthesized strand can be activated or deactivated, hence results in change in phenotype. And this phenotype, if it is favorable, it passes through the species by the process of natural selection. So this is all for today's video. In the next video, we will discuss about different steps of DNA replication in detail. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.